Okay, it is 12 o'clock, so why don't we go ahead and get started? I'd like to welcome everyone to the University of Colorado Medical Grand Rounds. I am incredibly pleased to welcome our speaker today, Dr. Esteban Burchard from the University of California, San Francisco. Before we get started, I'd like to just let everyone know there are no new Department of Medicine announcements for today. Just a reminder that all Department of Medicine Grand Rounds will stay virtual for at least the first half of the year. And then you can continue to receive CME and mock credit, which will be available to you in the chat. If you have questions for Dr. Burchard this afternoon, please use the Q&A feature. Myself and the chief medical residents will be monitoring that, and we will save and compile and ask all of the questions that we have time for at the end of Dr. Burchard's talk. And now on to our speaker. Dr. Esteban Burchard is the Hind Distinguished Professor of Medicine in Pharmaceutical Sciences in the Department of Bioengineering and Therapeutic Sciences in both the schools of pharmacy and medicine at UCSF. He did his undergraduate work at San Francisco State University. He was a medical student at Stanford, an intern and resident at the Brigham in Boston. He then did his pulmonary critical care fellowship back at UCSF and a number of graduate degrees at Stanford and Berkeley after that. He is the director, as we'll talk about, of the UCSF Asthma Collaboratory. He's also the director of the Pharmacology T32 program, the director of the Center for Genes, Environment, and Health at UCSF, and the co-director of the UCSF Career Development Program in the Omics of Lung Disease. He has been a member of President Obama's Precision Medicine Initiative and was on the advisory committee to the NIH. He has received a Lifetime Achievement Award from the American Thoracic Society, as well as in 2018, an Apple Teaching Award. He is a reviewer for medical journals around the world, including Pediatrics, Nature Genetics, the American Journal of Human Genetics, JAMA, as well as the New England Journal. He's also been featured in a number of popular press, including the Tavis Smiley Show and NPR Science Friday. He was also, interestingly, an All-American wrestler at SF State. I don't know if that factors into his current work, but we'll hear about that. And then what we're talking about today, the title of Dr. Burchard's talk is Race and Genetic Ancestry in Medicine, a Time for Reckoning and Racism. Dr. Burchard's work has focused primarily a part of his career on asthma, but also most recently in this issue of race and genetic ancestry and how it affects all of us and how it affects our patients. He is the director of this Asthma Collaboratory, as we spoke about, which is a large interdisciplinary research program focused on minority children. His team was the first to leverage genetic ancestry to identify novel genetic and environmental risk factors for lung disease and poor drug response. He is the principal investigator for the Asthma Translational Genomics Collaborative, the ATGC that you see listed on the first slide, which is the largest whole genome sequencing study of asthma in the world. In addition to his work with the federal government, he's also received a number of awards. There are too many to list. But I would like to recognize that he was, in 2018, recognized for a Lifetime Achievement Award for the National Medical Foundation, which is the nation's oldest Black medical association. And with all of that, I would like to very much welcome and thank our speaker today, Dr. Esteban Burchard.
Thank you, Dr. Connors, for the generous introduction. And good afternoon to all the interns and residents and students and faculty. It's an honor for me to be here. Been a long time fan and admirer of Dr. Schwartz and what you have done in Denver. And I thought long and hard about this talk and what I would have wanted to hear when I was an intern. So I'm going to begin with a little description of myself and then my journey. And for me as a resident, is really when my academic career took off. And so I want you guys to think, as you go through your internship and your residency, find a way to merge personal passion with academic rigor. If you could do that, you'll weather any storm that will come across you in your personal and professional life. And I was blessed to have a wonderful program director, Marshall Wolf, a wonderful mentorship during my second and third year residency, Jeff Drazen, and the two of them ignited a spark in me that changed my entire career. And it happened while I was a second and third year residents. So I hope that you guys can find the same good fortune that I did. So there's a huge debate in the nation and in medicine, and that's whether or not we should use race or ethnicity in clinical and biomedical research. There are really two camps. There's a camp that believes that race and ethnicity are purely social constructs, and there's camps that believe that genetic ancestry is a biologic construct. And the camps seem to be polarized, like this slide. But I've always believed, and many of us at UCSF believe, that it's really an interaction. And we had a paper in the New England Journal and invited into editorial in the New England Journal on January 6th. And I would encourage you to look that up. But less than 12 months ago, the chief clinical officer at UCSF came down with this hard email to all the faculty and staff saying that we as the UCSF, as a medical enterprise, are going to eliminate race ethnicity from all clinical tests. Now, that's well and good, but there is no discussion, there is no science behind this. It was just an emotional appeal to what was going on in the nation after George Floyd. Now, I've thought about race and ethnicity all my life. I am Mexican American. I come from a long line of farm workers. This is my grandfather, second from the right. And when I took this picture, I didn't realize that it was on top of my current issue of the New England Journal of Medicine. This is my family. You can see that we are mixed. This is my great-grandfather in the middle, my grandfather in the top right. You can see that we're racially mixed, a mixture of African, Native American, and European ancestry. And this is how I looked when I was in college. And I'm telling you this because we in medicine are having all these conversations about DEI training, microaggressions versus macroaggressions. And surprisingly, on my first day of medical school, when I looked like this, I was pulled over and frisked by the Stanford police while driving to my first medical school exam. That's a pretty macroaggression. And the problem is, I was with this gentleman, Dr. Rob Dodd, who is now a neurosurgeon. We were both pulled over, and it reminds me of an event that happened 54 years ago, in which Andrew Goodman, Mike Schwerner, and James Chaney were pulled over in Mississippi by the police and turned over to the KKK and killed. Now, that wasn't going to happen to me, but when you talk about microaggressions, this is pretty macro. And now Rob Dodd is a leading neurosurgeon at Stanford, and I'm a pulmonary and critical care physician. And when I was attending in the trauma unit at San Francisco General and the president of the United States came to San Francisco, the FBI would come to interview me. To think about these two guys being arrested and now being pretty senior leaders in academic medicine is a little bit surprising. So I come from, as I mentioned, a very diverse family. That's my mother in the middle and my wife and my children. We had our 23andMe done. That's my mother on the left. I'm on the right. I'm 27% Native American, a little bit African, and the rest is European. And this is a nice segue because now I'm going to talk about what I do for a living. So I study asthma. Asthma is the most common chronic lung disease in children in the world. And what was impressive to me is that it is a one disease that has the largest racial and ethnic disparity of any disease. You can see here on the right bar graph that the mortality rate is highest in African Americans and Puerto Ricans, but lowest in Mexicans. And early on, my mentor, Jeff Trazen, challenged me to think what proportion of this is due to genetics and what proportion of it is due to environmental factors. Well, when we started this work, we started looking around the world at how many genetic studies have included non-European populations. And to our surprise, this is Carlos Bustamante and Francisco de la Vega. We demonstrated that at the time, 96% of all modern genetic studies were performed in European populations, even though Europeans represent less than 12% of the world populations. And so instead of the Human Genome Project, we should have called it the European Genome Project. So that's a sidebar. But going back to my time as a resident, my mentor, Jeff Trazen, encouraged me to think big. And at the time, we were studying asthma severity in African Americans versus whites. And we identified a gene that was 40% more prevalent in African Americans compared to whites. And I reasoned that the racial differences between Latinos, Mexicans, and Puerto Ricans specifically was due to admixture. So we started a study called the Genetics of Asthma in Latino Americans, in which we wanted to recruit Latinos on the East Coast and compare them to Latinos in the West, in the South, in the Midwest, to look at genetic and environmental factors. I started this project when I was a senior resident, and I couldn't have done it by myself. And this is the value of mentorship. And to date, we have recruited over 13,000 well-characterized minority children. We collected very detailed information on genetics. We collected RNA. We have very detailed clinical measures, questionnaire measures on exposures and behaviors, perceived discrimination, and geocoded measures of air pollution. And we wanted to use these data to differentiate healthy individuals from sick individuals, and within children with asthma, what are the different flavors of asthma? Now, this is all well and good, but does it have clinical relevance? Because that's what we do at the end of the day. And one of the things that we did, which is very fortunate for me at the time, is we standardized spirometry across all the geographic centers in the United States, Mexico, and Puerto Rico. And this is an example of a young child blowing into a spirometer. He has a nose clip, and on the right, we have a depiction of airflow. On the top is exhalation, on the bottom is inspiration. And the dashed line is the flow volume of a child before getting an asthma medication. And the solid line on top is the difference in before and after. Now, this is very important, but you have to step back and ask, what are the determinants of lung function? Well, we know that sex is a big determinant of lung function. We know that age is a big determinant. We know that height is a big determinant. But what very few people talk about, and even as a pulmonary specialist, we don't talk about it, is race is a big determinant of lung function. And when I started my fellowship in 1998, we as a medical community, as a pulmonary community, we used a white standard reference equation for everyone. And when we had an African-American patient, we simply reduced the score by 15%. When we had Asian patients, we reduced the score by 8%. And this is what we considered race, what is considered race correction. Fortunately, the United States government recognized that the US is diverse. And so they started, the CDC started NHANES, National Health and Nutrition Examination Surveys, in which they sent buses, these sorts of buses, all over the nation. And these are clinical, mobile clinical units, and they sampled Caucasians. They called them Caucasians at the time. We call them now European Americans. They sampled Mexican Americans. They sampled African Americans. And they did multiple measures on height, lung function, blood pressure, all sorts of things. But when we look at lung function, what they found was this. On the y-axis, we have lung function. The higher you are, the better. On the x-axis, we have increase in age. We have males on the left, females on the right. And what you notice in a racial and ethnically stratified population survey, African Americans had lower levels of lung function compared to Mexicans and compared to whites. We know that lung function decreases at around age 25, and that's what you're seeing here. And it goes down for the rest of your life. And this is important. This is not race correction compared to what I said we did in 1998. So why does this matter? Well, that's who I am. That's just a reminder. This is my ancestry. When I was working in our pulmonary function lab as a brand new fellow, I had a gentleman come in who looked just like this man, President Obama. We all know his story. He's half African, half European. I had a firefighter who was half black and half white come in. He had an occupational injury. The insurance companies wanted to send him to an independent third party, which was the pulmonary lab at UCSF. And I was the pulmonary physician on working in the lab that day. And I watched the technician do the spirometry and then use what are called pull downs. So you decide whether or not the patient's black, white, or Asian. And since I grew up in the hood, I know how to code switch. I turned to my patient and I said, "Hey man, what are you? Are you a brother or are you white?" And he said, "Well, I'm half." And that really started my journey into taking genetic ancestry and applying it to clinical standards. We know that African Americans are racially mixed on average in the United States. And this graph on the right is a depiction of a sampling of about 275 African Americans who all self-identified as African American, who identified their 4 biologic grandparents as being 100% pure African American. But when we did the ancestry testing, just like what 23andMe does or Ancestry.com does, we can estimate what an individual's ancestral proportions are. And the average African American in the United States is 80% African, 20% European. But as you can see with President Obama, he's 50-50. So where does that lead us when we want to compare him to population averages? So my colleagues and one of my students, Max Seibold, who's now a professor here at National Jewish, asked the question, when we look at lung function, what should we use? Self-identified race versus genetic ancestry. And I'll just cut to the chase. This is what we found. We sampled hundreds and hundreds of healthy African Americans, adults, and children who had spirometry or lung function, and we had measures of genetic ancestry, and we just did a comparison. How does genetic ancestry correlate with lung function? And so what you see here, obviously, is an interaction. We have lung function on the y-axis. We have increasing amounts of African ancestry on the x-axis, and you can see that there's an interaction with increasing amounts of African ancestry. You have lower levels of lung function. And what we did is when we compared an average African American male to the standard reference values for Europeans or African Americans, we found that depending upon an individual's genetic ancestry, we would either underestimate their lung function or overestimate their lung function. Now, to our knowledge, we were the first group in the world to publish this. We published this in the New England Journal. And I want to clarify that this is not race correction. This is ancestry adjusted. We went on to repeat these experiments in Mexico using Mexican children and indigenous Native American group or Native American groups. And this project was led by Chris Ginyu and Andres Moreno Estrada. So Chris is one of my first graduate students and one of my best. And he's currently a faculty there at the University of Colorado, Denver. And he's soon on his way to be probably one of the next world leaders in translational genetics. And what Chris and his colleagues demonstrated that depending on the type of Native American ancestry you had, it changed your lung function by as much as 10%. In the previous example in the New England Journal paper, we demonstrated that we can improve the diagnosis of lung function by as much as 15% if we use genetic ancestry. So now in 2 independent populations, African Americans from the United States and Mexicans from Mexico, incorporating Native American ancestry, we saw significant differences in lung function. So that wasn't enough. We published that paper in 2014. The big question was, do social and environmental factors mediate or confound the relationship between genetic ancestry and lung function? And so since we had all the data, we were able to ask this question, and this is led by Maria Pino-Yanez and Nita Tucker, who was a pulmonary fellow in my group at the time. And we asked a simple question: what's driving this association with genetic ancestry and lung function? We measured this in 5,500 children. We had data on environmental exposures. They're listed on the left. We had data on socioeconomic factors listed there on the right. And we asked, what's the biggest driver? And what Maria and Nita demonstrated is that even after adjusting for these social and environmental factors, that genetic ancestry was the biggest driver. And this is how it looked when we did the similar plots from our New England Journal paper and the science papers. Again, with increasing amounts of African ancestry in Latinos now, we had lower levels of lung function. Now, this is very important because what I'm referring to is disease misclassification. For the average person who's getting spirometry, it probably doesn't make a difference. But if you are a firefighter, occupational, who has occupational risk factors, and you get referred to a pulmonary function lab, it's pretty serious. And it's pretty serious that it's pretty important that we get the diagnosis right. And if we have any degree of misclassification, it leads to inappropriate referrals, inappropriate tests, treatments, or delayed treatments. It has tremendous implications for disability ratings, lung transplant referrals, preoperative risk, workers' compensation, and reimbursement for pulmonary rehabilitation. Now, these are just a simple short list of implications, clinical implications for lung disease. You could take this same thing and talk about kidney function and when not to start dialysis, when and when not to refer for transplant. The same is true for lung function. Now, you've heard a lot of opinions. Should we drop race and ethnicity from kidney function or lung function or all of clinical medicine? And I want you to know that it's time to balance the scales. Overwhelming majority of opinions on this are commentaries or viewpoints. Very little is based on empiric evidence. I demonstrated and presented to you empiric evidence that race, ethnicity, and genetic ancestry are important in lung function. Now, it's very important that we recognize the danger of untrained personalities. Everyone's saying that they have an opinion, they're an expert. And that's what this is. Mike Lindell, who's a recovering drug addict, crack addict, and alcoholic, who's now become the My Pillow guy. Now he goes around saying, trust me. And this has real life implications. And this is the danger of the bully pulpit. This is the paper that was published recently in the American Journal of Public Health, comparing the number of anti-Asian hashtags to the calendar year from the time when President Trump first used the term China virus or Kung flu virus in a tweet. And you can see the number of anti-Asian hashtag hits for anti-Asian tweets go up dramatically. I'm not making this up. This is recently published in March of 2020 or March of 2021. Now I want to switch gears, and I want to. I'm mindful of time, but I do want to talk about the big elephant in the room. And you know that we've had a JAMA controversy. The editor of JAMA, Journal of the American Medical Association, was fired. And it came from an interview he did with Ms. Katz, the director of public health for New York City. And here are two white guys bantering about, and they basically agreed that no physician is racist. And so there's no racism in medicine. Well, this is a problem. There's a tremendous backlash because we know that from the national events that there are racist attitudes in the United States, and it is pervasive in every aspect of our life, including medicine. Now, this required people to look at the demographics of who makes the decisions on what gets published in major medical journals. And this is the demographic breakdown of the editorial staff at JAMA and the New England Journal. And you see that for JAMA, at least, the majority are white, very few Latinos, very few African Americans, and Asians. This is a further breakdown of JAMA by the top editors, the deputy editors, the senior associate editors, and consulting editors. And this is how it looks when we looked at the top 100 cardiology journals. 16% of the editorial staff are women. The majority are white men. This is the same is true for NIH study sections. And why this is important is these are the gatekeepers. What you and I read every day as our gospel, the medical literature. I read the New England Journal on a weekly basis. I read JAMA, especially during the COVID epidemic. But if we have biases of what gets published and what doesn't get published, if we have biases in what gets funded and what doesn't get funded, then we have a problem. It's not an egalitarian system. And going back to what racial bias in medicine looks like, in the early days of the COVID pandemic, this group out of Mount Sinai in New York published this paper in JAMA saying that the disparity in African Americans with COVID was due to the simple one gene. We know that's not true. We have a paper in the Journal of American Heart Association that criticized programs to increase diversity in medicine. That paper was retracted. We had a paper published in the New England Journal on the biases of pulse oximeters, which is a key tool for all of medicine, but pulmonary medicine during the time of COVID. So it's inappropriate to blame the high rates of COVID on a single gene in African American children. It's inappropriate in this day when we are America, with the rich diversity that we have, to say that programs to increase diversity are harming medicine. Claiming that we haven't done the hard work to measure pulse oximeters across different populations with different skin tones. We need to do that work. We need empirical evidence. And so the consequences of throwing out race and ethnicity is problematic. You don't want to throw the baby out with the bathwater, because if we resort to a race-blind approach, we will default to a European standard. We need a clinical and scientific alternative. There are dangerous consequences for not educating ourselves before speaking out. So where do we go from here? Well, race is still used in some medical measurements, although there's a fight. As I said in the beginning of my talk, the chief medical officer at UCSF stopped the use of race, ethnicity for all kidney function and lung function without any empiric data. I think it's important that we use it. I think that going forward, and this is what we discussed in our January 6th, 2021 New England Journal piece, that race, ethnicity is still important, and we should augment it with genetic ancestry. In the next 10 years, I think that genetic ancestry and ancestral diversity will be important for finding rare genetic variants that are causing disease. The most important thing is the epidemiologic importance of race, ethnicity will never disappear because it's a wonderful epidemiologic proxy for what people experience in their own personal lives, in their communities, and out there in the real world. So I'm going to sum it up. I think that race, genetic ancestry, and medicine are intertwined. Race-specific measures are not the same as race-corrected measures. What we say when we have the bully pulpit can have dangerous consequences. And then finally, the epidemiologic importance of race and ethnicity will never go away. I want to thank many people that helped contribute to this body of work. I've had many wonderful mentors along the way. I mentioned that I started this project when I was a senior resident, took a year off and worked with Jeff Drazen and Ed Silverman, came to UCSF and was mentored by Neil Risch. And I've had some wonderful collaborators, Elad Ziv, Noah Zeitlin, Louisa Burrell, and that's my team there on the bottom left corner. I want to thank all my collaborators nationwide and our funding agency. And with that, I'm happy to open it up for questions. Thank you for the opportunity to come, and thank you for your attention.
Esteban, thank you so much for that presentation. I'm going to turn it over to Jeff to ask some specific questions that have come into the chat box, but I just wanted to make a statement and ask a more general question before we get to the other queries after your talk. First of all, I want to congratulate you on being a national, international leader in this area. Secondly, I want everyone to know how important this is. It's important not only for what we do in medicine in terms of understanding disease etiology and treating people effectively, diagnosing people precisely, but it's important for how we understand how medicine fits into the communities of individuals that we take care of and the responsibility that we have to those communities. And I think it's absolutely critical to understanding the complex nature of the etiology and treatment of many of the problems that we deal with as internists. So my question to you, Esteban, is something that may be difficult for you to answer, is how, what's your estimate in terms of how big a factor this is and how much we're missing the mark in terms of our ability to both use our laboratory tests to diagnose disease and also to understand disease etiology. What are we missing? What are we missing 90% or are we missing 10%? Where are you on that spectrum?
I believe that we're missing a significant amount of disease. So go back to my firefighter who had an on-the-job inhalational injury, smoke injury. As a pulmonary physician, we know that that'll lead to long-term emphysema, pulmonary decline, oxygen dependency. So rightfully so, he wanted to get disability and he should have got disability. And if there's an error in the measurement by as much as 15% for African Americans, that error could either put you on the side where you won't qualify for disability benefits, or could put you on the side where you do qualify. So that's an example that affects millions of people worldwide. Right now, in the United States, if you're Asian and you get your lungs measured, you're going to be compared to a white reference group. So we don't know the proportion of damage that we're incurring there. For kidney function, delayed time to start transplant or delayed time to dialysis is important. We just had a beautiful paper come out yesterday in JACI. The main Journal of Allergy and Clinical Immunology is actually selected as the best paper of the month, in which we demonstrated that the current clinical standards for deciding on whether to start children with moderate to severe asthma on one of the new asthma biologics is dependent upon peripheral blood cell counts. Well, peripheral blood cell counts vary by race, ethnicity. And so we, using our data of 13,000 children, we demonstrated that amongst the African-American children with moderate to severe asthma, 50% would not be eligible for that medication, dupilumab in particular, based on blood cell counts. 27% of Puerto Ricans would not be eligible. It goes on and on and on. We know that white blood cell counts vary by race, ethnicity, yet we make a decision every single day when to start chemo based upon your ANC. So that is based on a European standard, and I think I think we have to have an empiric look at how we measure people and compare them to population averages.
Great. Thank you. Jeff?
Yeah, thanks, David, and thanks, Dr. Burchard. There are a lot of questions, as you can imagine. I'm going to have the first question come from our friends at National Jewish and one of our leaders there in pulmonary medicine and in racial disparity. Her question is, when do you think, given all that you talked about and all that we know, it's still appropriate to use race, and in what context? And is it the patient's perception of their race, or their phenotypes and their genetics, or some combination of both that we should use?
I think it's a combination of both. And that's what we articulated in our New England Journal piece on January 6th. Race, so let me answer it this way. We know that epigenetic variation is influenced by environmental factors. Dr. Schwartz has demonstrated that in mice. And we know that it's influenced by genetic factors. Using our data set, we published a beautiful paper. We simply asked the question, what proportion of the variation in epigenetics is explained by genetic ancestry or social and environmental factors that co-vary with self-identified race? And what we demonstrated in two independent populations of Puerto Ricans and Mexicans, that 75% of the variation in epigenetics is driven by genetic ancestry. 25% is driven by factors that co-vary with self-identified race, ethnicity. That could be environmental factors, that could be experiences of discrimination, socioeconomic status, dietary factors. And so we published that in eLife, and it was, the senior author on that was Noah Zeitlin, who's featured there, and Josh Gallanter from my lab. We published that in eLife, and it was the first empiric example of it's a combination of the two.
That's great, thank you. There are a couple questions here. You use the term African ancestry or African American ancestry. How is that defined? Because obviously the African continent is very heterogeneous. Is this self-defined? Is this ancestrally defined, or some other way?
Well, it's a moving target. We know that all population is derived from Africa, that Africa is the most genetically diverse population continent in the world. There's a significant amount of intra-variation within African populations. The United States happens to have been populated by populations from West Africa. That's the majority. It's a moving target. We have in the old days, in the beginning of the Human Genome Project, we had a small sampling of African populations, but now they have this thing called H3 Africa, in which they're doing a comprehensive sampling of all of Africa to get better reference panels. So the short answer is self-defined, but it's also driven by existing databases and what's out there now.
And there's a related question that takes a slightly different spin, which is, if we know that those with more African-American ancestry have lower pulmonary function tests, and you show that pretty clearly, does this actually put them at higher risk of lung disease or complications from lung disease?
No. So it just happens to be that they're lower than Mexicans and Europeans, but that's not the right comparison. The comparison is African Americans to African Americans. That's the right comparison. So to put it simple, for those of us that had children, when we bring our children to the pediatrician, if I told you that little Johnny's at the 75th percentile for height, you obviously know that Johnny's taller than 75% of boys his age and shorter than 25%. But what you don't know is, are we comparing him to a Vietnamese person, our population average, or a Samoan average, or East African average? And so it's really important that we have appropriate reference populations to do comparisons to.
And then maybe a related question to that, because there are so many of these domains, as you mentioned, that don't have these appropriate ancestral comparisons, how do you use race and ethnicity in your current clinical practice?
It's first of all, I don't think we should be using a race blind approach. I and Neil Poe and others are leading the fight for lung function, and Neil's doing it for kidney function. Within our own pulmonary community, the American Thoracic Society, even amongst experts who are trained in pulmonary medicine, there's a debate on whether we should have a race blind approach to lung function or race-based. And we're advocating to go the next step, which is to include genetic ancestry. And I think ultimately, with genetic testing, the cost of genetic testing going down so precipitously, that it'll be easier to implement in the clinic.
Esteban, discuss the complications or the extent to which you'd be able to include genetic ancestry. In other words, how many markers, how cumbersome would this be? What are the expenses related to this? Just to put it in context for everyone listening.
Well, we haven't done a cost-effective analysis yet, but I think that's the next step. Many people in an audience have probably had their ancestry measured by 23andMe or Ancestry.com. It's a simple saliva test. It's a simple gene chip. It costs about less than $50. For important decisions like when to start transplant proceedings or not, I think it makes a big difference. I think that we would be foolish if we stayed stagnant in what we know today in medicine and not incorporate the knowledge that we've gained from the Human Genome Project. We'd be foolish. And more disease genes have been identified probably in the last 10 years than the entire history of modern human genetics. And you know it's important when you have a tumor, and the first thing they're going to do is they're going to sequence that tumor to identify whether or not you're eligible for novel pharmacogenetic targets. So we are already doing it, and the field of oncology is leading the way. And no one thinks twice about the cost of sequencing a tumor.
Esteban, as we think about the use of personalized medicine and making this as precise as possible, I think one of the concerns raised in the clinics, the questions here, is that often people of disadvantaged backgrounds have less access to personalized medicine. And of course, as things, as that becomes a more wide field and costs come down, that should or could improve. But how do you assure that the most economically disadvantaged groups have access to these same treatments and therapies as we base those therapies more on personalized medicine?
That's an excellent point. And that was the point of our JACI paper. In the last 5 years, there have been some wonderful new drugs for asthma. They're not curative, but they work very, very, very well. Dupilumab is the one that is really having a significant impact on the field. Well, if you're not insured or you're in a state that doesn't have dupilumab on the Medicaid, Medicare formulary, the cost of dupilumab is $48,000 per year. That's higher than the average annual minimum wage salary for most states in the United States and our territories in Puerto Rico. So the first thing is identifying the problem. I'm doing my part as an activist physician that has a social agenda to illuminate the problems to the world. How do we deal with the problems is left up to this generation of interns and residents.
I love it. And maybe on that activist idea, there's two questions that are related to that. The first is the most specific. We'll start there. How do you advocate for increasing racial diversity in patients who are recruited for clinical trials?
We published a paper on this in PLOS Medicine. We had expressed our opinions on how to do this. It's multifactorial. We need diversity in the scientists. We need diversity in the physicians that are doing the work, in part because they're most likely to go out and target their own populations. They're most likely to be accepted by their own communities. They're most likely to understand the nuances of communities. I have to confess that when I was a resident at Boston, which is very racially divided town, very racially divided medical system, that my mentors didn't know what a Hispanic was, didn't know the difference between Mexicans and Puerto Ricans. And this is where diversity came in, because when I saw the initial data describing the disparity of asthma of East Coast Hispanics versus the West Coast Hispanics, it helped that I'm Mexican, have lived on both coasts, and understood that there's a difference between Puerto Ricans and Mexicans, not only culturally, but genetically. Puerto Ricans tend to have more African ancestry, less Native American ancestry. Mexicans, on average, are 50% Native American, with little African ancestry. And so that was the aha moment for me when I was standing around my mentors, Jeff Drazen, Ed Silverman. These are heavyweights in medical education. Jeff was the former editor of New England Journal of Medicine. And when I looked at data, I saw a different solution than when they looked at the data. And that's based on diversity.
And a related question that may be harder and or more interesting, and I know this comes from our chief residents and from our residents. How do you propose that clinicians take actual action to avoid using race-based medicine that is not evidence-based? And what recommendations do you provide to trainees who want to combat misinformation, but run into the busy life of a trainee and or pushback from their healthcare systems?
There's a paper that came out in June of 2020 in the New England Journal. The first author was Vyas, talking about race, ethnicity. I think the title was Hidden in Plain Sight. And they went down a whole list of clinical parameters in which they evaluated was race appropriate. And I would agree with them that in some cases it's not. There's no evidence. It's all anecdotal. There's a paper published in PNAS from the University of Virginia, which medical students and medical residents all thought that African Americans had a higher pain tolerance and dulled nerve endings, and therefore had a higher pain tolerance. So that kind of stuff has to be ferreted out. We have to do the hard work. We have to clean our own house. And that's why I'm privileged to speak to this brand new class of interns that are only on the job now for 3 months, and residents. And they're going to be the ones that challenge us, push us, challenge the status quo, and do your due diligence and homework.
And I put the link to the Vyas paper in the chat there for the audience so they can take a look at that if they haven't seen it. To put a finer point on it, if one of those interns comes into my office tomorrow as the program director, or they come into your office tomorrow and they say, what can I do now? How would you help advise that person?
It's not an easy fix. What we can do now is to educate ourselves. I really have had a wonderful journey in my career, and it really started when I was a resident. I was in a medical scientist program at Stanford. It was not an MD PhD, but it was like an MD PhD light, where I got to spend my first 3 years of medical school in a genetics lab, actually, looking at heart, lung transplant rejection and gene expressions. I wasn't turned on by that, but when I fell in love when I got to the Brigham and started working with my mentor, Jeff Drazen. And even though I had great training, I got more training and got more training. I went to Berkeley and got an MPH. I went to Stanford and did a year with Neil Risch. I got a degree at the Harvard School of Public Health. So I educated myself. I didn't just stop. And medicine is an art and a science. It's not a trade. It's filled with continuous learning, and we have to challenge ourselves to continue to educate ourselves and stay ahead of the curve. And for the interns and residents out there, this is probably one of the most wonderful times of your life, where you're like a sponge, and you could just learn so much, and you'll see so much and do so much. And you don't clock in, you don't clock out. You live with this. This is us.
Thank you. As program director and audience member, I appreciate that. Different line of questioning here. We have a couple minutes left. Do you have any sense, what are the biologic mechanisms for these ancestry-based differences in lung function, independent of height and sex and things like that?
Well, there have been several. I didn't show any genes, and that's for more scientific talk. Underlying these ancestral differences are probably genetic, are positive genes and genetic variants that will explain a certain proportion of the variation in lung function or height. We know that, and that those genetic variants differ by population. Some will be common to all populations, and some will be private. So, for example, Elad, who's featured on my slide, demonstrated that and published this in the journal Nature, that breast cancer's prevalence is higher in whites and lowest in Native Americans. Mexicans are in between, and he identified and demonstrated that women who have inherited a Native American estrogen receptor were protected from developing breast cancer compared to those Mexicans that inherited a European receptor. We know that PCSK9 was identified in African Americans, but when they dug in on the biology, they found that the drug works whether you're black, white, purple, blue, or pink. And so what I'm arguing for is that we use diversity and leverage it to scientific advantage.
I think the last question, unless David has anything he wants to add at the end, and Esteban, as both of us are pulmonologists, we know that there are large clinical trial groups that exist in single countries. There's large Brazilian groups, there's large Australian groups, et cetera. And there's also all the work that is just sort of European-centric over time. How do you currently generalize those results to populations other than those that are studied when you're working in your clinical practice?
You can't. And I have a piece, an invited editorial coming out in New England Journal, probably in October, where we discuss this. If we did a study in Brazil, the results will be specific for Brazil, not necessarily generalizable. We always try to fit a round hole in a square peg, or, you know, for medicine, and we just hope that results are generalizable, but they're not always. And in the case of the asthma medications, in the paper that I said came out yesterday, we demonstrated clearly that 50% of African-American children with moderate to severe asthma don't qualify based on blood parameters. And so the company that made that medication just assumed that what they found in whites would be generalizable. It doesn't work that way. We are a very rich and diverse country. More than half the United States children are minorities now. Latinos make up about 18% of the population. African Americans make up about 15%. We're increasingly becoming more and more diverse. And we need to embrace that and make sure that that our clinical trials represent the diversity of America. I think we could do it. And as far, go ahead.
And I would just add, and that our approach to understanding disease and our approach to patient care take into account the diversity of the patients.
And I want you to, as a resident and interns, think about physician activist. Think about Dr. Fauci and all that he has risked, his personal life, the life of his family, his career, but he has stood up for public health. in a tremendously positive way. It's a great role model.
Dr. Burchard, it is unfortunately 1 o'clock. I think we could do this for another hour. I just want to say, on behalf of all the University of Colorado, a sincere thank you. You spoke obviously beautifully and eloquently about a very complex topic, and it's one that's close to all of our hearts, and it's one that's going to influence the future of medicine. Thank you for addressing so much of it to our interns and to our residents and to our faculty. We appreciate your time today. Thank you again.
Thank you.